So again, I want to say I'm recording this. I don't want you to say your names. I'm not going to say your name. So again, you're saying to me that you were confused about a lot of questions on the test, and that the figures were the biggest thing that you remember you were confused about. Okay.
I only took the first one. The second one I wasn't here for, but the first one I just didn't understand like the back page, the little graph.
The graphs or the figures?
Figures.
The figures. So you were confused about like the molecules and which molecule was which. And,
yeah, I didn't know what it looks like.
You don't know what it looks like. And why is that? Like, why do you think that happened? Now the three of you, have anyone else experienced this before? So you pretty much everybody was the same thing with the figures. Why do you think that was? I mean, I went through it. Thank you. I went through it and explained what it was. I drew them on the board for you. I gave you several different options, right? I gave you some practice problems with them. I even gave you a quick, right, remember the day before the test, I gave you guys a quick review, a summary, and we went through and identified each of those. So why is it that you think you got confused on being able to identify, right? Identify the molecules. What confused you? What can you improve for next time? Because I'm assuming some of you, if not most of you, want to retake the test, right? Mm-hmm. So what are you going to do? What went wrong the first two times, right? Because you did it twice, and they were pretty much the same figures. What went wrong the first two times that led to that confusion? Maybe it was texting in class. Maybe that was something. Give me some ideas. What?
Well, I just didn't know some of the parts. I knew some of it. I just didn't know.
You knew some of it, but I mean, whether you knew some of it or none of it, or you were completely confused, what can you do? I'll start. What I'm doing is I'm going back and doing kind of a small group tutoring session with you to go back and look at the questions and see what you were confused about. That's what I'm doing as a teacher. I'm asking you, what are you going to do? What are you doing? What went wrong the first time that we can improve? If we don't share with each other, if we're not honest with ourselves, we're not going to be able to improve. So what went wrong the first two times? Any ideas?
I think it's just hard for the figures because like all of them have the hydrogen and the carbon inside of it. So when I see the letters, it just looks like the same thing, but just like in a different form.
They all kind of blend together.
Yeah, they all kind of look the same.
Yeah.
So I don't know which one is which.
Okay. Why don't we all pay attention though? Why don't we all be on the same page at least? Let's start with that because there's only a few of us in here. And so here's the next question. Did you make index cards where you drew the general form of the figure and listed on the back the name and the characteristics that were going to be the clue for your brain to say, that's DNA, that's RNA, that's protein, Did you do that? No. See, that's it, man. There it is. I don't think, I want to say, let me see them. Let me see the index cards. You don't have them. Well, show them to me on Monday because I want to see these figures. Because it's hard to me, and then if you did, and they're correct, because I had someone, I think I shared with a couple classes, I had someone show me their index cards, and you know they were wrong? They had labeled the wrong molecules. Can you believe that? They drew a protein, and they had put DNA on the back of the card, and they drew something that I don't even know what it was, and they labeled it a lipid. Like they had the hydrogens connected to hydrogens. It was kind of a crazy thing. So somebody was just trying, you know what that was? That wasn't really, I don't know if she studied those. She said she studied those cards, but I'm not sure if she studied them or if she just made the cards quickly so that she could write in the blue sheets that she did cards. And she didn't think I'd look at them, you know what I'm saying? So I'm not sure if that's what happened, but whatever. I can't believe, and when we go through it today, I can't believe that if you made cards and they were accurate and you practiced them every day, like you got up in the morning, you went over them, and you went over them with a friend at lunch or advisory, then you went over them again at night, and you did that for 4 or 5 days, I cannot believe you would not be able to know what these molecules are. I mean, I can't believe you couldn't look at something and say, that's DNA. At least know it's a nucleic acid, right? And that's a protein, and that's a peptide bond. Like the peptide bond's probably the hardest one to tell, because like you were saying, it's actually a bond in between all that. But if you know if you had on the back of an index card protein or polypeptide, and then on the back of the card you said peptide bond holds the monomers together, I can't believe you couldn't put that in your brain, right? I just think, to me, and if you find that hard to do, then we have to have a deeper discussion. We have to dive deeper into the cause of that, because that shouldn't be happening, right? Do we agree? I mean, this has been like 2 weeks, 2 tests. We've had a video out that explained the first test. I've identified, I've drawn it like, I want to say 10 times on the board. At this point, if you're having problems identifying the molecule, and what I hear from most of you, and I thank you for being honest, is I think I hear from most of you that you just didn't do the index cards and that's it. We're done. But if you're doing the index cards, you're having problems, then like I said, maybe this stuff's wrong, or maybe there's some other issue we have to delve deeper into. All right, so let's just go ahead. And I heard that there were some other questions like you got confused about. All right, so I think we've at least identified one strategy. We really need to make those index cards and really like, okay, I got to know these shapes. I have to know this. I have to know this, so I better put on index cards. And that's going to happen all year long. There's a ton of stuff you're going to have to just memorize. The steps to protein synthesis. But that wasn't all there was to the test, right? It wasn't just memorization. You had to actually apply it, right? And do you think that that's why it was so confusing to you, some of those questions? Because they weren't straightforward memorization questions, right? If they were straightforward memorization questions, I think a lot of people would have done better the first time. So let's go and start with the questions one at a time and see what we're talking about here. The first question says, which of the following is not a characteristic of life? Does everybody know the characteristics of life, the 7 or 8 characteristics of life? When you're looking at something, you look at a bunny rabbit, you say it's alive. You look at a rock and you say it's not alive. Why?
They don't react.
Well, that's right. That's one. Somebody learned from the test. If they got it right, great. If they got it wrong, they sure learned from the test, because it doesn't react to the environment. You can poke a rock all day long and it doesn't react, right? And it doesn't process energy. It'll absorb light, energy, heat, and it'll get hot and hot and cold and cold. It'll take up heat and lose it. But it can't store energy, right? A rock doesn't multiply. Do you agree? Mm-hmm. You don't get like a rock growing. You can leave a rock on a table, and then tomorrow you come back, there's 4 rocks on your table, right? I mean, it doesn't happen all by itself. They don't have sex, right? They don't reproduce in any way. Rocks don't do the things that living things do, right? But what are those things? That's the question. What were those things that we talked about? They don't have cells, that's right. I like that you're looking it up on your, I see you, right? You're reading it up. Are you just putting it from your memory? That's good that you know it off the top of your head. If you don't know these 8 things off the top of your head, then this is something you got to put in your head. You have to ask yourself, and isn't this like a basic understanding of the world? What is alive and what isn't? Mm-hmm. To say, I just know, that's not right. I've had kids that didn't think trees were alive. They didn't know plants were alive. They said, plants are alive? Because you know why? Because they thought that, and it's not that we all laugh sometimes when you hear stuff like that, but it's like, trees don't move, right? And in their mind, things that are alive moved. But we know that that's not necessarily true, right? We know that there's 8 characteristics that make something alive. They have cells, right? We know that they process energy, they grow, they reproduce, they evolve, right? They respond to their environment. They have complex chemical reactions as part of that whole processing energy. And what else? Anything? I have 6. That was 6. I mentioned, what? React. I mentioned both of those. That's good. Yeah? Regulation. They regulate their temperature. They conduct homeostasis, right? Living things keep, either by behavior or internally, they keep a set point for almost every trait that they have. pH, blood glucose, whatever, right? So see how we listed all those 7? Do you see how I listed them? You should be able to do that. If you're not able to do that at this point, you another index card. Or 7 index cards. Or you know what? Some people don't do index cards. Fine. Write it out 5 or 10 times, 20 times. Quiz yourself. I had a friend, as I shared with you in class, I had a friend in med school that would put all the information. He had glow-in-the-dark paint, you can't see it in the day, but you turn off the lights and you see it. He wrote all the symbols on the roof, on I don't know how he did it, but on the ceiling. I'm not telling you to do this. I'm just telling you that he was innovative. He felt like he was an artist, so he said he'd do that. And he wrote it all on the ceiling, and every time he'd go to bed, he'd spend like an hour going over it, and when he'd wake up, he'd go over it, and the lights were off, so there's only one thing he could look at. So I thought that was really innovative. That worked for him. I didn't do that. I just used index cards, or I wrote it out 5 or 6 times. I have a colleague here, I think some of you may know her, Miss Gabriel. We took master's classes together, and what she would do is she would rewrite everything. She'd write it all out. I used to think, man, that's crazy. How are you going to write all that out over and over again? But that worked for her. And so there's 100 ways you can do this. You can do it on Quizlet cards, right? You can do it any way you want. But if you need help, you need help in figuring out how to do it, come and see me. But you got to get these things in your mind. It should be quick. 7 things in life. That's a protein, that's DNA, that's this. I'm not asking you to identify the type and where and what and whatever. I just need you to know when you're looking at a molecule, that's what it is. All right, so which of the following is not a characteristic of life? It says being multicellular. Are there multicellular living things? Mm-hmm. Sure are. We're one of them. But is all life multicellular? No, it's not. And that's why this one's wrong. So even though, and this got a lot of people, and I can understand why they got it wrong the first time, because you see, there is multicellular life, so you say that's a characteristic, but that's not a characteristic of all life, right? So regulation sure is, right? Oh wait, I scratched it out as if it's not the answer. That is the answer, because the word is what? Not. The word is not. So which of these is not a characteristic of life? It's being multicellular, right? So which is regulation? Is regulation one? Yeah. Oh, you don't see it. I'm sorry. Let me pause it. All right, there, it's up on the board now. So which of these is not a characteristic of life, right? And so that's why being multicellular was the answer. That word not gets a lot of people. So you have to be careful reading questions. This is kind of a test-taking skill. So regulation is certainly one of them, right? We all agree on that. So because it is a characteristic of life, it's not the answer. What about responding to the environment? That is a characteristic of life, that's right. So that's not the answer. Do we all agree on that? And adaptation to the environment, that's environmental changes. It's called evolution, by the way, right? That's one of the ones that we didn't say. No one said it earlier. So evolution. Everybody okay with that? The idea that species evolve to adapt to the environment. So that's not an answer because it is a characteristic of life. So again, I want to emphasize these are characteristics. Did anybody mess this one up? Did anybody feel, again, don't say your name, just raise your hand or nod. Did anybody get this one wrong because they reversed the not, right? Okay, so now that's a really good, easy way of not making a mistake again. Watch for the not. So all life does this in one way or another. Now, different forms of life do it different ways, but they all do it. But not all life is multicellular, and that's why that one's not the answer. All right. A person eats 3 meals a day and gains 4 kilojoules of energy, through a series of chemical reactions. Then they go for a run and lose 50 kilojoules of energy. After you sum up the total, the person lost 10 kilojoules of energy. What is the final result called? Metabolism. And this was confusing to a lot of people because what they thought is like, what's kilojoules, right? What is kilojoules? How does that have to do anything? There was one key word that should have triggered your brain. If you studied enough, if you studied enough and you went over enough, there should have been one word that kind of triggered it. Some, or maybe a couple. Energy, sum, boom, metabolism. Because that's what metabolism is. It's the sum of the... Now here, this one is your negative, your loss of energy, loss... Right? And here's where your gain, anabolism's gain. So that's fine, and food is what you eat. You don't need to know catabolism and anabolism, right? But what you need to know is metabolism, right?
If you reverse
This
it
is
on
so.
the next test, don't we have to know what
No, I wouldn't do that to you. I'll just come up with another scenario. I'll have to sit down and think about it, where it's going to ask you again the sum of. If I tell you something, I'm not going to test something. I'm not going to test it. I may throw catabolism and anabolism there again as a choice, answer choice. Maybe I'll put something else. But I'm not gonna ask you the definition of those if I've told you don't need to know it. That wouldn't be fair, and you'd have a right to complain about that, and I would probably change it.
When do we retake?
Once we're done with this, and if you feel like you want to retake it, I have to write the test again. I have to sit down and write it. I could probably write it this weekend if you guys want to take it on Monday. So when someone who is not a diabetic eats sugar, her blood glucose goes up and then comes down. And then I gave you a little chart here. I thought that was a nice little chart. And see, the blood glucose is here, and she eats something and then comes back down, right? This is a normal, like this happens to anybody who's not a diabetic. So if their blood glucose goes too low, you see it went too low, and the liver releases glucose also through something called glucagon, but you'll learn about that later. It's called a feedback mechanism. So one goes down, there's a negative feedback that tells it to go back up. So liver releases glucose, keeping blood glucose within a range of about 70 to 99. So somewhere within here, you have blood glucose going up and down. Right after meals, glucose may go above 100. See how it went above 100? But it never goes, if you're not diabetic, it's not going to go above 140. So when you eat a candy bar, you eat a big meal, you're going to see your blood, if you were monitoring your blood glucose, it would go up, but it just wouldn't go above 140. Now, did you need to understand all that? Kind of, you need to understand the up and down, but really you don't need to know much about blood glucose to answer the question. The above graph to the right is that of blood glucose changes in a non-diabetic person's blood sugar during the day. When a diabetic's blood glucose gets above 99, they have to take insulin to bring it back down. Oh man, sorry about that. I wrote it at 2 in the morning. So what is the process of keeping the blood glucose within this range called, right? So the idea that you have this normal range, we call it normal because it's the average, normal range, right? What do we call that? Right, hemostasis. All right. It's homeostasis, but no, because this is not asking you what is it called. See, that was the trick. You had to read. Are my test questions asking you to read the questions and interpret and think through the problems? Absolutely, because if you don't, you're going to get it wrong. Why do I do that? Not because I want to make it tricky, but because that's the way ACT, SAT, and end of course exams are written. And so you need to be able to process the information. The question, if you read it carefully, says, what is the process of keeping the blood glucose range within a range? Well, what's the process called of keeping it in here? It didn't ask, in the first test question I asked you the first time, I said, what would happen if you didn't regulate it and it just kept going up? That's what I asked you. And that would be what would happen in the end of that would be death, right? There's only one answer. It's homeostasis. Oh, I'm sorry, there's not. There's more than one. There is more than one answer. You're confusing me. Hold on a second. So it's not death, all right? So it's not, death is not the answer.
Hibernate.
Homeostasis, hibernating is what some organisms do. Some mammals do. There are some frogs, different organisms do to get through a rough time. The winter, in the desert, some frogs will dig themselves underground and hibernate for 14 years before they come back out. Locusts are famous for that, right? They come out every 14 years. Well, though they don't really hibernate, I don't think.
What if somebody was digging and then
they would if they were digging and they were there, they would find the frog, right? So hibernation is not the answer, all right? Now, what is homeostasis? What's the characteristic of life that homeostasis falls under?
Regulation.
Regulation. We literally just listed regulation in problem number one. We're at problem 3, and I ask you, this is homeostasis, keeping something at a set point. That is the definition of homeostasis, and people didn't choose homeostasis. They remember they didn't choose regulation. People chose homeostasis, right, because they remembered the up and down deal with the homeostasis, but they didn't understand what regulation was. And so the key is, here's the key. Stop trying to identify key words in a quiz and say, oh, it's regulation, it's this, that. When he says this, I'm going to say that. Stop trying to follow that test-taking strategy. That's good to take sometimes when your back's against the wall and you have no other choice, you don't remember, then do that. But the only way to do well on my tests and or end-of-course exams and ACTs is to actually understand the process. You have to understand what regulation is. If you didn't understand what regulation was, then you need to ask those questions in class or come for tutoring. So let me ask this question really quickly. Do you know what regulation is? No. Okay. So the example I gave you, if you go back and read and look at the video of all the different times we mentioned in class, regulation, oh my God. You know I didn't record any of that? Dang, I'm not recording that. It's already repeated. So I went through and discussed all the different ways that we could tell the difference between the 4 molecules. If you're confused, go back and look at the original video on it. I'm sorry that I didn't record it. All right, so now we're going to look at the figures. Now we're going to go ahead and use the questions to answer. We're going to use these to answer the question. And luckily I can move this here and look at it. And we can use our knowledge of the previous slide to answer these. It says figure 2. Which one's figure 2? Well, this one's figure 2, right? Figure 2 represents which of the following. Choose all that apply. We identified this as a what? Protein. And what do we identify and a polypeptide? What is a polypeptide versus a protein? Why is it both?
Because it's two because the bond holds it together.
That's right, because there's many peptide bonds, right? Right, so it's a polypeptide and it's a protein. It's not a chain, and what else is it, rather? It's also a what? It's a chain of amino acids. So it's all 3. It is choose all they apply. And so let's look at it. And we looked at it in the previous slide, but I'll do it again here. We have amino acid 1, amino acid 2, right? Amino acid 3, all connected by peptide bonds. Amino acid 4. So amino acid 1, do you see that? Amino acid 2, amino acid 3. So we call that a chain, right? Do you agree? So it's a chain of amino acids connected by peptide bonds. I can live with that. So that's it. And of course, polypeptide means it's more than one peptide bond, many peptide bonds. So here's column A. It says column A. That was my mistake. I made that correction after the first class took it. And I actually made it in the first class that took it. That was my mistake. Again, changing a question from the previous test. So in A, in figure 1, represents the part of the molecule that holds the monomers together. You didn't have to look at it. What's the part of DNA, of nucleic acids, that holds the monomers together? Phosphate sugar, phosphate sugar, phosphate sugar. And let me show you. Look, what is it? What's holding? What are the monomers? The monomers, this one, let's circle the monomer, square off the monomers like we did in the protein. 1 monomer, 2 monomer, 3 monomer, right? 4 monomer, 5 monomer. Do you see that? 1 nucleotide. So A, or rather T, A, C, T, C. There's 1, 2, 3, 4, 5 monomers. And what holds these together is the phosphate sugar backbone. That's this thing. Also, I pointed it here. Do you see? It's this yellow one. So however you want to look at it. Again, back to it, you need to develop your skill of reading a diagram. Can a diagram be confusing? For sure it can. That's why you got to memorize your landmarks, right? That's why you've got to memorize the parts that will help you identify it. You're going to see this again this year. You'll see it possibly in the end of course exam, but I can almost guarantee you'll see it in AP Biology. I can guarantee you're going to see it in HBS. I can guarantee that you're going to see it, you're probably going to see it again on your ACT when you're trying to fight for a college scholarship. And you will definitely see it in college when you get to biology there. And almost everybody takes high school or college biology. Because very, very rarely do you find someone who's not a science major that tries to take chemistry or physics. But you have to take a science. All right, so there it is. That's those two. So the answer, as I said, as we all agreed, was sugar phosphate backbone, right? All right, so figure 1, there's an attractive force between the two strands. And what two strands are we talking about? And this is the one that was given to you, right? This is strand 1, and it's again, there's two directions. This is strand 1, right? And here's strand 2 over here, right? Do you see that? All right, so two strands that kind of wind around each other, they stick to each other. We're going to unzip them. When we do DNA replication, we're going to unzip them. In HBS, when you do electrophoresis and PCR, you're going to unzip them. You're actually going to unzip them. If you think of two Velcro strips, one Velcro strip is one strand, the other Velcro strip's the other. So they stick together. These stick together like Velcro, okay, except it's hydrogen bonding and not hook and loop. So here you have one strand here, another strand here. What holds these two together? Again, I made the mistake and I left the answer on there for you. And so if you weren't able to read that or didn't read that, again, that's back to the skill, test-taking skill. All right, so you should know that hydrogen bonds hold the two together. So what is the attractive force between the strands at position D? What is that force called? This hydrogen bonds. That's it. That's all there is to it. Now, I don't know about you, but I think if you knew the structures of these molecules, you knew your 7 or 8 characteristics of life, you knew your hierarchy of life, if you knew those 3 things, I would expect 100% so far. Maybe you mess up 1 or 2, right? Because you misread it. But I wouldn't expect people to get 30 or 40% if they knew those things that we went over for 2 weeks many, many different ways. All right. So figure 2 shows a molecule CHON and sometimes has sulfur. We already know it's protein, right? We identified as such, but even if you see that there's sulfur in it, what did we say about carbohydrates? What do carbohydrates have? Equal amounts of what? Oxygen and carbon. And what are the only 3 things that carbohydrates have? Carbon, hydrogen, oxygen, CHO, or however you want to say it. That's right. What do we know about lipids? What do lipids mainly have? Carbon and hydrogen, there's some oxygen. And some lipids have all kinds of crazy other things, but for the most part, that's what we got. Now look at protein. Now DNA has what? Carbon, hydrogen, oxygen, all that's true. But then it has what? It has nitrogen, here the nitrogenous space, that's correct, you don't see it, but they do, they have nitrogen there. And what else do you see? It's right in your face. It's right in your face. Well, hydrogen bonds, but what element is, what's the other element? It's right in your face. The P. Do you see every phosphate? Phosphorus, the element's phosphorus. Phosphate is phosphorus with 4 oxygens. So we know that this one, if we just would have memorized the elements in each of these, just that. If you can't look at this and see the geometric difference, the shapes are different, you should know that this is P, O, N, H, right? For sure. Oh, yeah, sorry. Thank you. There should definitely be a C. All these have C's, which is why we call this organic, right? Because they all have C's. Very good. And this has what? C, H, O, N, sometimes S, right? Sometimes sulfur. And you can see the S. Do you all see the S? It's right there. It's one of the side chains. Then this one here, there's only three elements here. C, H, O. That's it. Boom. All right. So, I had someone tell me that this had an S in it on this test. I was confused. And what about this one? It's mostly C and H. That's the whole point about lipids. That's why they're nonpolar. That's why they don't mix with water. Because they just don't mix. Because oil and water doesn't mix, right? Because they're made mostly of carbon and hydrogen. That's a nonpolar molecule. And sometimes they have, and of course they have some oxygen, and then sometimes they have other things as well. But most of this is going to be C and H. That's the key to lipids. Now let's go back to the question. Figure 2 shows a molecule made of CHON, sometimes sulfur. In the figure, what are the bonds that hold the units together? Peptide bonds. They're called polypeptides for a reason. The answer is peptide bonds. Okay. Figure 3 shows a molecule that's made mostly of carbon, oxygen, and hydrogen. Do we need to look at it real quick? Carbon, oxygen. Here's figure 3. Do you see it? What is it? Quick. Carbohydrates. So let's look at this again. Made mostly of carbon, oxygen, and hydrogen. The number of carbon and oxygen are about the same. Wow. I said that like 5 times, and you guys were able to read it. If you knew that, just that much, what would you have been able to answer without even looking at the picture? Carbohydrate. You didn't even need the picture. Let's say you have a problem with pictures, but it doesn't matter. If you knew that carbohydrates are just carbon, hydrogen, and oxygen, the number of carbons equals the number of hydrogens, I state the definition, you should have got that right. We agree? I mean, if you disagree, please, I have no problem with you saying I don't agree. Figure 4 is a molecule that has 3 long chains of carbon and hydrogen. Long chains. Now what are you thinking long chains of carbon and hydrogen? Lipid. You're thinking lipid. Maybe not. Let's finish reading the thing. But definitely long chain carbon and hydrogen. I'm thinking lipid. I'm ready to go. I'm ready to jump. And I've only read the first sentence. In fact, the whole molecule has less than... Am I recording this? Yeah, okay, good. Shoot. In fact, the whole molecule has less oxygen than either carbon or hydrogen. Now you even have some more information. It's less oxygen than carbon or hydrogen. 2 of the chains has all the bonds of carbon filled. What do we call that when a lipid has all its carbon bonds attached to hydrogen or another carbon? What do we call that? Hydrogen bond, right? When there's no double bond. Not hydrogen bond. I don't know what I'm saying. I was expecting someone to give me the right answer. So we have, what do we call it? Maybe a picture will help, even though pictures. What do we call it when we have all the carbon? There's only single bonds. They're all the available carbon bonds have hydrogens on them. What do we call it? I'll give you another few seconds to think about it. It's something we covered a couple times. You heard the words a lot in your life. I'm sure you've heard this. Say what? Well, they certainly are organic, but that's not what I'm looking for. The thing when you see this is there's no double bonds. I hear there's double bonds. There's a difference between these 2 and this one, right? These 2 are saturated. That's right. And this is what? Unsaturated. Remember that whole idea about saturated and unsaturated fats, saturated and unsaturated lipids. You actually didn't have to know that for the test, but that's why it was another clue to tell you what this was. That's all. I was trying to give you clues. I said, I'm sorry, I didn't want to say saturated or unsaturated because that would be too easy, but I just kind of said these are double bonds and the others don't have any bonds. So if you would have heard saturated and unsaturated, hopefully you would have quickly jumped on lipid, but Yeah, the answer is lipid.
Wait.
What type of molecule is it? It's a lipid. Why is it a lipid? Because it's mostly carbon and hydrogen, because the whole molecule has less oxygen than either one of the other two, and and you have both saturated and unsaturated types. So those are all clues as to what it was. It was lipids. And that's the test. That's the test. I'm going to send this off to you. If you want a copy, I don't think you need a copy. You can take you can take a copy of the test, although I threw most of them away because nobody wanted them. Are we good? So if you retake the test, you better